So this chapter will focus on the structure and functioning of membranes found in the plasma membrane and membranes of membrane-bound organelles like the mitochondria you see here, and there's rough endoplasmic reticula, probably right there. And so we're going to look at the first section here, and your learning outcomes are going to be to describe the components of biological membranes and to explain the fluid mosaic model of membrane structure. So over here to the right, we have an image of the plasma membrane we saw back in the prior chapter. It's a double layer of phospholipids, and in the middle is going to be these nonpolar part. And that nonpolar part is made of fatty acid tails that are hydrocarbons. And when you're nonpolar, that means you're hydrophobic. You don't like water. And if you're outside the cell out here, so let's say we're extracellular here, and then here we're inside in the cytoplasm, and you're intracellular, and here it's water-based environment. So it's a polar environment on either side. So these tails are kind of pushed inwards, and then the heads on the outside here, those are polar. So you have polar head on the outside and polar on the inside. And these phospholipids, they're free to move around, and embedded within there are going to be these proteins that can also move around. So overall, this if they're able to move freely, and by the way, the proteins will only move around freely if they are not tied to the cytoskeleton. So like right here in the images here, you'll see that these proteins, they are tied or tethered to some of the cytoskeleton within, so they can't move around as freely, but others are not, like this protein right here. And some other proteins that may have sugars tied to them. Those can move around freely and randomly, and they refer to this as a fluid mosaic model. And the fluid means that it can move around. So they compare these, sometimes these proteins, like kind of ships in the sea floating around there. And so taking a look here, the phospholipids are arranged in a bilayer. The proteins are globular, and globular means that they're not fibers. Like here on the outside in the extracellular matrix that we see out here of the cell, we see protein fibers. And those protein fibers basically are not soluble in the fluid environment. That means they don't dissolve in there, but a globular protein does. So these are the membrane proteins are going to be globular proteins, and they are basically at home within the plasma membrane, so they're sort of dissolved within that environment, or mixed well within there. So this is an image from chapter 3, looking at the basic structure of one of those phospholipids. We'll represent it with a polar head right here, and then the nonpolar tails there. Structural formula here itself. The phospholipid will have these fatty acid tails, and they may be saturated or unsaturated. Keep in mind here, something to remember, that if you are unsaturated, that means you're going to have a double bond. There's a little double bond in there. It looks like it's got an equal sign there. And anywhere there's a double bond, that causes that hydrocarbon chain to bend. Okay, and that's going to be important when we talk about how fluid the membrane is, because if all of the nonpolar tails here are all straight, and that means they're saturated, just like you see here, then these molecules can pack more tightly together, and you're more likely to be less fluid or more solid. And so the more kinks you have or bends, the more fluid you will be. So there's variation depending on the conditions that the cell experiences, whether it's a cold environment or warm environment. You can see here it's basically, if you took a triglyceride, which is a fat or an oil, you would have a glycerol molecule which has 3 carbons, 1, 2, 3, and on 2 of the carbons is where you have your fatty acid tails. Triglyceride would have 3, but for a phospholipid, one of them is basically replaced by a phosphate group and another group called choline. And the phosphate and the choline are polar, so this is going to make this part of the molecule polar and this part over here nonpolar. So polar and nonpolar, and there's a fancy word for that that's called amphipathic. And amphi, kind of like amphibian, dual nature, water, land kind of thing here, dual nature, polar, nonpolar. You remember that polar is going to be considered hydrophilic. It's polar. It's like water. It can associate with water quite nicely. And non-polar would be considered hydrophobic, which means water-fearing. So here's that model again of the bilayer. Study it. It would be great if you could sketch a basic membrane and label some of the things you see in here, like glycoproteins. Those are important. And then you're going to have these integral proteins that go integral. They're integrated within the membrane here. They have those special properties. And then you're going to have peripheral proteins that are off just to the inside of the membrane there. Notice that you also have a molecule called cholesterol. And these cholesterols we looked at back in chapter 3, and they're basically 1, 2, 3, 4 ring a lipid structure, and so they're right at home within the nonpolar region within the bilayer. And the cholesterol plays a role in helping to maintain the stability of the membrane. So the components of the membrane we've already mentioned, and so this is why I think it would be a great idea if you can do a basic sketch and label some of these important parts. So you're going to have, of course, phospholipids arranged in the bilayer with the nonpolar tails to the inside. It's fluid. Then you're going to have transmembrane proteins. And transmembrane proteins are going to be proteins that can go all the way across the membrane. Generally, a transmembrane protein is a specific type of protein called an integral protein. Okay, so the transmembrane is specific in that it goes all the way through. You're going to see images in your studies of membranes where some of these proteins, there's not one drawn in here, but some of them are only partially integrated in. They don't go all the way through. So that makes it an integral protein, but it's not a transmembrane. Transmembrane goes all the way across. So there's a difference there. So both are integral because they go into the membrane to some extent, but a transmembrane goes all the way through. Okay. So we might say crosses all the way through. So all transmembrane proteins are integral proteins, but not all integral proteins are transmembrane. Is the idea there. So there's going to be some of these peripheral proteins that are off to the inside here. And as I mentioned earlier, they connect to the cytoskeleton. And then you might have some cell surface markers here that give your cells identity, help cells recognize each other. And then they also help your body, for example, recognize cells that belong to you and those that do not belong to you, self from non-self. And so some of these molecules are proteins. The proteins generally in this image, this diagram here, are colored a bluish color. Lipids look more golden color. But so you can see here there's a protein right there, and it has this short chain of sugars. So we might say a short chain is oligos, which means many, but which means some. That's not the same as poly, which means many. I messed up right now. So it's poly. Oligo means some. So they're oligosaccharides, which means a some. Okay. Or several, but not many like poly. And so these sugar molecules, and some of them are attached, the sugar molecules are attached to glycolipids also as well. So you have glycoprotein and you have glycolipid, and this overall is going to give this sort of a sugar coating around the entire cell, and this gives the cell a uniqueness when it comes to specific cell types. So one cell type has a different kind of fingerprint, so to speak, of the sugar covering. And so the glycoproteins and glycolipids are collectively called and by the way, glyco refers to sugar or being sweet. And so it's glycocalyx is the term used, and calyx is a covering. Okay. So here is, there's a scanning electron micrograph of the phospholipid bilayer. And if you're going to really study the structure a bit more closely, you might use an electron microscope. So EM stands for electron microscope, and we briefly mentioned those in our chapter on cells, prior chapter. There's a transmission electron microscope, and that's abbreviated TEM, and then scanning. And recall that the scanning electron microscope will scan the surface and produces a three-dimensional image, whereas a transmission, you have a very thin section through the specimen, and then you shoot electrons through there, and it gives you a two-dimensional image. So what you have here on this image right here, you can see the scale is on the order of nanometers. 25 nanometers is what this bar here to give it scale. This is a transmission electron microscope, and it is two-dimensional. They color enhance it. It's so you can see some of the structures here. This would be inside, this blue area would be inside the cytoplasm of one cell, and this is another cell. So there's two cells that are near each other, and you can see the phospholipid bilayer there, and they colored it in orange. So you can see there's two layers there. And so a really cool image. And then another way to study the membrane is called freeze fracture, and this would involve scanning and produce a three-dimensional image. So the basic idea here is get a cell or some tissue with cells, and you're going to embed it within a medium, some kind of medium that's going to sort of solidify. So it might be some sort of an epoxy or something. And then you get a sharp blade and you slice into that medium and you split it. And when you split it, this medium is going to basically pull on the surface of the plasma membrane, and typically the plasma membrane will split the two layers apart. In other words, it'll split along where the fatty acid tails are, the nonpolar tails. So you can see when you're taking off that piece of that medium here, the top layer pulls off partially, and that leaves the bottom layer. And so some proteins are going to go with the top layer. Remember, the integral proteins are within there, and some are going to stay on the bottom. So you can see that here in the second image right here. And you can see some of the proteins stayed with the top layer and some stayed on the bottom. Now, once you do that, now you're going to coat it with a metal, because you need to coat metal to give contrast in electron microscopy. There's no light here, so it's not stains or dyes. You coat it with a membrane, and then you use a scanning electron micrograph and you hit it with electrons, and you get this image here. Here, both in this area right here, both membranes are still there, so you can see the surface of the membrane and you can see some proteins there. And then this is the inner layer where this part right here got pulled off. And so you can see some of the proteins, those little bumps in there. And so you get this three-dimensional surface here. And so that was produced by a scanning electron microscope. So in section 2, we're going to look at the phospholipids themselves, which are the basic foundation of the membrane. And so your learning outcomes here are going to be to list the different components of a phospholipid. So if you're going to be able to list them, you got to visualize them. So again, I recommend sketching and labeling, and explain how the membranes, how they're going to form spontaneously in structure. And then describe the factors involved in membrane fluidity, just determining how fluid it is. So recall that the structure of the phospholipid consists of a glycerol, which is a 3-carbon polyalcohol. And so we were to sketch that. Alcohols have hydroxyl group. It's going to be 3 carbons. This is how the glycerol starts. And polyalcohol just means it has more than one OH. It's the hydroxyl group. A simple alcohol would have one OH. And all the other carbons are just going to be your basic hydrocarbon molecule, which is filled in with hydrogens. Remember that a carbon has to satisfy the octet rule, so each dash is a pair of electrons. So there's your glycerol, and then right here where the hydroxyls are, that's where the fatty acids would be tied to. So in this diagram here, this is where your glycerol is, okay? And so there's the oxygen for one of the OHs and the other one. And so you're going to have 2 fatty acid tails attached. And the fatty acid part, this part right here, is nonpolar. And so that nonpolar means that it's going to be hydrophobic, water-fearing, okay? And then you're going to have your phosphate group. And the phosphate group actually has the phosphate, which is one of the functional groups to know. And then there's another part of it called choline, which I mentioned earlier. So the phosphate group, because it's polar, is going to be hydrophilic, water-loving. And so remember, we're going to draw it more simply here as just a sphere with little tails. And here, instead of drawing the membrane going left to right, they drew it sort of going up and down, and you can see those tails end up spontaneously moving inward and the heads to the outside of the membrane, and or to the outsides. And on one side is the inside of the cell, on the other side is the outside of the cell, which is a water-based environment. So this is a more favorable situation that the tails, since they fear water, they move towards the inside. And anything that's dissolved in the water that's out here is gonna be hydrophilic as well, and would rather, or it would be more favorable to be with the water. This has to do with the stability of the system. And it's more energetically stable if polar is with polar. So if water is a polar environment, polar molecules would be out here, and so with the polar heads would be out here, and the tails would be more. It would be a more stable situation for them to move inwards. And in this case here, these polar molecules would have a hard time crossing through because it's going to be hard to go through the nonpolar parts. So this is going to make what the plasma membrane is as being a barrier that helps sort of guard against just anything moving back and forth between the cell. So this is going to make allow plasma membrane to regulate what gets in and out of the cell, which is where we're moving once we look at the structure here. Again, the way those phospholipids form is spontaneous. That means it's going to be more favorable for the system here. Your hydrocarbon tails are going to be towards the inside, and we can see that in this drawing here. They're toward the inside, those tails in there. And the polar heads are going to move outward where you have your polar environment, water-based environment. So the phospholipid bilayer is fluid. That was mentioned earlier. We will refer to as a fluid mosaic model. And the basically, because the water on the inside and the outside of the cell, this is going to help maintain that bilayer structure because it's just more energetically favorable for those polar heads to be facing outward and the nonpolar tails facing inward. And so the phospholipid are free to move around, and so it's referred to as fluid. And any proteins that are not attached to any of the cytoskeleton inside can also move around, and they refer to that as the fluid mosaic model. And you can see evidence of this if you were to combine two different cells. Here's a human cell, and it's going to be identifiable as a human cell because the proteins that are in the membrane are characteristic of human type proteins. The mouse would have similar proteins and similar functioning, but you would be able to distinguish overall the components there. There's especially things like glycoproteins and so on. And then here you have your mouse cell. And if you were to combine the two cells, the two membranes can fuse because it is fluid in situation. So as they fuse together, those proteins that are human and mouse, if given enough time, begin to spread out and move about randomly. And so this is indeed seen, and this provides evidence for that fluidness or the fluid mosaic model that we have been talking about. Now, what influences fluidity? This was the third objective for this section here. One is going to be whether or not you have a saturated or unsaturated fats, the level of the saturated fats. Remember the saturated fats, if we were to take a phospholipid that had saturated, the tails are going to be straight. Okay? But if you got unsaturated, the tails are going to be bent, right? And so if the tails are straight, then phospholipids can pack together a lot easier, and that tends to make the membrane less fluid. Okay, it starts to become more solid instead. But if we're like this, it's hard. If we're like the phospholipid here, it's harder for the next phospholipid to come near it because that bend is kind of keeping the other phospholipid away, so that makes it more fluid. As you can see the comparison here in these two membranes here. This membrane here has a lot of unsaturated fatty acids, a lot of kinks or bends in the fatty acid tail. So this one would be more fluid. If we look at this membrane right here, those fatty acids tend to be saturated and they're straighter. You can see the cholesterol molecules within there too. Now, which one is going to be more fluid? The one on the top, if we just looked at the amount of unsaturated fatty acids that are in there. Okay, so it's the amount of unsaturated that's in there. The more unsaturated fatty acids, the more fluid you'll be. Conversely, if you look at it from the other way, the more saturated fatty acids, the less fluid it would be, which is how it's worded here. It's just two different ways of saying the same thing, and it's why we want to make sure we don't memorize things word for word. We want to be careful of how things are stated. There's another factor that can influence fluidity, and that one is temperature. As you know, when temperature goes up, the kinetic energies of the particles themselves begin to increase, and that means their speed at which they move increases. Right? In solids, they start to vibrate faster. You get enough energy, it melts, and the molecules start moving more freely. Same thing here. If you have warmer temperature, then your membrane is going to be more fluid, right? So if you're cooler, less fluid. So the membrane has to have characteristics that depend on the temperature that these cells are going to be in. And an example here would be if you're a bacterial cell, and you live in a cold environment, you need to maintain the fluidity of the membrane for it to function correctly. So a bacteria that lives in a cold environment is going to have more unsaturated, more of those types that have the bend in the membrane, in the fatty acid tails. And that keeps those phospholipids from packing together closely, and it'll keep it more fluid at colder temperatures, right? The opposite would be true if you are in a warmer environment. You're probably going to want to have more saturated fatty acids. Then it also depends on where the membrane is. If we look more closely at the diagram up here, they were actually comparing the bilayer membrane, because the membrane-bound organelles like Golgi bodies and Golgi apparatus, endoplasmic reticulum, are made of phospholipid bilayers too, and this one is of an endoplasmic reticulum. Rough ER, smooth ER, one of those. And the one down here is an actual plasma membrane. Okay? So the plasma membrane tends to have more saturated fatty acids. Okay? And that means the phospholipids pack together more closely and would be somewhat a little bit less fluid. But when you look at the endoplasmic reticulum, there is more unsaturated fatty acids in here, and so the packing of those molecules is less, so it's more fluid in there. And you also see that the fatty acid tails tend to be shorter in the organelles, so your membranes tend to be thinner, and in the plasma membrane tends to be thicker. And you can see that here with this integral. This is an integral protein. It goes, and it's a transmembrane protein because it goes all the way across. And you can see here the distance here going all the way across is about 20 amino acids, and this one is 25. So the plasma membrane tends to be thicker, and the membranes of organelles tend to be thinner. So generalizing, organelles tend to have less packing of the phospholipids because there's more unsaturated fatty acids. You can see in the diagram because of the bends, the fatty acid tails. And for the plasma membrane, fatty acid tails are going to be longer and tend to have less of those unsaturated fatty acid tails. And so they would be a little more stable, a little less fluid. Now notice you have a lot more cholesterol molecules within this membrane here than you do here. And the cholesterol helps maintain the fluidity and the stability of the plasma membrane. So in this section 3, we're going to look at proteins specifically, the different types of proteins you find in the plasma membrane. So your learning outcomes here are going to be to illustrate the functions of the membrane proteins. That means just be able to describe them and what they do, and then illustrate how proteins can associate with the membrane. How are they actually placed within the membrane? And then your third objective is to identify transmembrane domains. So when we look at proteins of the membrane, they do have proteins in general have quite variable functions. But for the membrane, some of them serve as transporters. They help transport things across the membrane because things that are typically outside or inside the cell are in a water-based environment. They're hydrophilic. And if that's the case, they're going to have a hard time going through the membrane because the inside of the membrane is hydrophobic. Some of them serve as enzymes, and enzymes help run metabolism. These are globular proteins that attach to the reactants of a reaction going on within the cell, and then help promote that reaction or speed up the reaction. Some of them serve as receptors. Some of them serve as those identity markers, part of that glycocalyx we mentioned earlier. Some of them are adhesive in nature and help bind cells to cells, and then some of them help attach or are attached to the cytoskeleton for perhaps anchoring. And so here we have all three of these illustrated. Here's a transport protein. You can see it's a pore. This is going to allow some solute particles to enter through the plasma membrane. These are likely polar molecules or ions, both of which can dissolve nicely in a water-based environment. And they're going to have a hard time crossing through this hydrophobic region. So hydrophobic or nonpolar, they just basically not going to be able to get through. All right, so that's a no-no. So instead, you have specific proteins that allow for the transport through there. So the plasma membrane regulates what gets in or out. Here you have enzyme as a function here, and this is illustrated here just diagrammatic in diagram form here. This diamond here just represents some reactant that's going to go through a chemical change, and it fits perfectly with this membrane protein that's serving as an enzyme. And so it goes and attaches here, and then the enzyme causes the chemical reaction to occur. That's part of your metabolism, and out comes a new product. And they're just symbolizing that with a sphere. It's something different, something new. Some of them serve as receptors, and you may be a receptor for some sort of chemical messenger. Like in our bodies, we have hormones. So this is a chemical, and it binds to a specific site on the receptor. The receptor is specific for whatever it's receiving here. In this case, could be some sort of message molecule. And when it binds, this is a globular protein. It's going to change the shape. It doesn't go through the membrane, but it changes the shape, and the shape change on this side actually sends a message. And so this is going to be like, looks like Wi-Fi or something, right? Like it's sending a message inward that causes changes to occur within the cells. So in this case here, the receptors can serve to receive information from the outside. And then here you have a glycoprotein on the bottom, so that's part of the identity. You can see the little short chain saccharide there. And then for adhesion, cell to cell, so here you see two cells. There's the plasma membrane of one on the left. Here's a plasma membrane of one on the right. And here there's some sort of adhesion or cell junction going on there. And then some serve as attachment to the cytoskeleton. So you can see here those look like those microfilaments made of the two chains of actin, fibrous protein found in the cytoskeleton. When it comes to proteins, there's the diverse functions, and these functions are sort of course, related to their functioning overall. When you have peripheral proteins, okay, those are going to be off to the side, and some of these proteins are going to have anchoring molecules, okay. And these are going to be special molecules that are illustrated on the next one here, and this is the anchoring molecule that you see right here. And when it comes to the anchoring molecules, you can see here that the protein is out here to the outside. It's of the membrane here. And you're going to have some sort of a modified lipid, and that's right there. And they colored it a little different from the rest of the phospholipids. So this modified lipid here has, of course, a nonpolar component to it. So it's quite at home there in this nonpolar hydrophobic region right here. And in fact, if it tried to come out, it would not want to be out there because this is a polar environment out here. Okay, this is nonpolar. And polar likes water, so this is a hydrophilic environment. And so then this anchoring molecule is going to have a component to it that helps attach to the protein over here. Okay, so these would be your anchoring molecules. So the anchoring molecule is going to have that nonpolar region. That's that part right there that sits within there. And then it'll have some sort of bonding domain to it, right about there, that binds to the rest of your protein. And so now we move on to these integral proteins, and those that are going to span. Integral just means it's at least part of the way into the membrane. So if we did our bilayer right here, okay, and you had a protein that went part of the way in, it's an integral protein because it's integrated within there. And if you had another protein out here that went all the way across, it's also an integral protein. Okay. But it's a transmembrane protein because it goes all the way across. Okay, so here's the bigger point, though, is that if you are going to look at these transmembrane proteins, they have to have polar and nonpolar natures to them. So we were looking at the amino acids that make up proteins. There was 20 of them. And while I didn't expect anyone to remember the structures of all 20, one of the things that we should be able to recall is that the amino acid themselves, their structure, okay, they have a central carbon with a hydrogen there. They're going to have a carboxyl group out here, and they're going to have an amino group out here. Okay, and right here is your variable R group, and that R group, some R groups are nonpolar and some are polar. So if you're going to be a part of that protein, that's an integral protein, where it's blue right here, right there, you're going to have a great deal of nonpolar R side groups, this part right here, to make up that part of the protein, and that allows it to be embedded nicely within the plasma membrane. Whereas out here, you're going to have more either polar or ionic side chains. There's also ionic side chains as well. And that allows it to associate with the polar heads and the polar extracellular environment and the polar internal cellular environment here. So that's what this slide is talking about when it comes to integral proteins. They're going to have nonpolar regions that are going to be embedded on the interior of the bilayer. That's the blue area there. Whereas the polar regions are going to be protruding onto the sides, which is, I guess this is an orange color right here. And so you can see this would be the polar parts of it, right? That's the rough structure of these types of proteins. And now this is important here because this is a fluid situation, and this protein is free to move around. And if the direction is going to move around is outward or inward, when it moves outward, that water-based environment is going to push that nonpolar part back in because the nonpolar part and the water are not going to mix very well. And so the water molecules that are on the outside and the inside are more stable being with each other because of their hydrogen bonding, whereas the nonpolar is not very stable out there, so that water wanting to be with itself basically pushes the nonpolar part back in. Okay? So it's not just going to go floating around out freely from there. So this sort of helps keep those proteins within there. And so this is a rather simple integral protein. Here's one protein that's folded back and forth on each other in its tertiary structure overall. And you can see it has several parts that go through the plasma membrane that are nonpolar right there. And then you're going to have pores within the membrane. And I went ahead and found an illustration. Remember that in the secondary structure of proteins, the primary structure is going to be the just basically the sequence of amino acids, and this is dictated by the gene for that protein. The instructions are making it. It tells you the order to put the amino acid, and the protein begins to fold to form a secondary structure. And that secondary structure produces two basic kinds: helical, which is twisted, and then beta pleated sheets or folded sheets. Okay, and this would be helical right here. These ones here. These were similar to those integral proteins we were just looking at earlier. So those are helices, and the part that's in the membrane is going to be nonpolar, right? But when it comes to making pores, and these pores are little holes, little openings in the membrane. and they serve as channels for specific molecules. So there may be, for example, a sodium channel produced by a pore-making protein. These pore-making proteins tend to be cylinders made of beta sheets. So these sheets are those beta-pleated sheets, right? And so you can see the sheets right here. There's a sheet and there's another sheet, and it says a beta-pleated sheeting here, and then right in there is the pore. And that pore these pores are usually specific channels for specific things. I mentioned earlier, like for a sodium channel, for example. And the thing about this is that the way the protein is folded is that the interior of it, the hollow space within, is going to be made of amino acids that have those polar R groups, side chains. So they face inward. So that means that out here, you have a hydrophilic environment because the water and other molecules out here is a polar environment. It's probably better to say polar environment. And if you have a polar nature on the inside of this beta barrel, they call them. Okay, so that cylinder is called a beta barrel. On the inside, then that means water and any components can move through here quite nicely to the other side, where we also have a polar environment on the other side of the membrane. Now, the outside of the protein here, the outside of it would be having those non-polar side groups for those amino acids. So the protein folds in a way in which the polar side groups go inward into the barrel and the non-polar outward, so that this part of the protein that's right around here on there has that hydrophobic nature and keeps that protein embedded within the membrane there. So these are structures called beta barrels. So there was no illustration in your lab manual, so I went and found one to share with what a beta barrel might look like. And they help form these pores.